done your transformation, normalization of your data, you can visualize your data. Just create a principal component, do a principal component analysis, and your raw data. So you will ignore this, okay, when you are on PCA, and then just proceed, because automatically it takes the first column of your data. But luckily it happened to be the treatment of my interest. So it will. Something happened for you?
Not yet.
Okay, cool. So it's still working. If you click on proceed, you can see here. Let me. Maybe it's a little bit bigger. Okay. This is a byte plot. Okay, a byte plot, coordination plot, whatever you name it. But this is an extraction of eigenvectors and eigenvalues from this PCA, principal coordinate analysis, okay? Which tries to basically break down the multidimensionality into a linear dimension, okay? A 2D dimension. And then what you can see here, you need to read this line, these numbers, okay? These numbers are your eigenvalues and the coordination that define the coordination of each sample in this space. See what happens? We remove the variance coming from that, and now you have those beautifully separated clusters. Let me increase the size here a little bit. You see what I mean? And if you also want to have them a little bit more visualized, you can add this ellipsis down here. And then voila. This is beautiful. Use it if you know that there are some confounding factors in your data and you don't want their effect. You can do distance-based redundancy analysis or redundancy analysis. Okay? Good. Now we are done with this. Now we know that treatment definitely has an effect on this microgram data. Okay? You can see the separation, but now we want to dig a little bit deeper. What causes this separation over here? What causes these basically samples to cluster, group together, but group distant from the other one? Okay, we do that with differential Abban analysis, and you can see it's already here. But before that, you can also take a look at this bar plot, stack bar plot. Everybody, this is the distribution. I mean, across all the samples. But also, it shows you colors those stack bar plot of each species. So let's show you some cool trick here. So let's say I'm gonna download this as it is, okay? Before I do further. Let's not do that now. But because this naming, you see, there are a lot of species that their annotation is not complete. They are known to, let's say, genus level, but their species is uncultured, whatever. It makes it very long and unreadable. What we do here, we set this to genus level already, okay? And then instead of sample, we select a variable treatment, okay? And then here, we pass on weeks. Again, this weeks I know is important, okay? And then recreate the plot and see how it looks. I'll tell you. So you can add a grid as well here. So let's say if you had another variable, like let's say 6 or something. So you could create a multifaceted with different rows in the grid as well, like this, okay? But this one needs to be empty. Delete this, and then you just say, I don't know, I don't think I have anything else to show you, but that has enough levels. But anyways, that's one thing. I try to show all the possible options, Masa, if I answer you. Otherwise, you can download the table down here. Okay, I'm going to differential Abban analysis. Do I still see these changes in Provatella? So what I do here, I select the taxa level for agglomeration. Let's do species. Let's do genus, because I'm interested. This plot was in genus to see if it can capture anything. Okay, let's go to genus. Here you have different modelers. You have DSEG2 and COM-BC, an intercept, okay, when the levels of these are 0, you have nothing, you still have something, okay? And then here it gives you the estimate for treatment group, protozoa versus naive. So it's important, the order of this, for us to be able to read the following plots, okay? This is protozoa versus naive. This little text here, and then paste it into this name of your desired model extract. Set your alpha level to the default whatever, and then let's just set this color as it is. And down here you have waterfall plot, volcano, and heat map. Let's just choose the waterfall plot as the default, and then click start analysis. And was it this guy?
Yes.
There we go. And it's protozoa versus naive. Okay? And again, I'm not sure if this is the correct result. Why? Because if you go back to this your stack bar plot, you can see it that it's only in week 0 that you have higher protozoa. So week is still confounding. Okay? That's why DSIG is not super powerful. We need to go for something that can basically disentangle this effect of unwanted variables. Well, let's say that we have some sort of rough estimate of what's going up and down and how to read this plot. This is log 2 full chain. What is log 2 full chain? So, what is the division of 6/3 is 2, right? What if it's the division of 3/10 is some decimal proportions, okay? When we are presenting these results, we don't say we have increased protella, we say we have higher protella. It doesn't mean it has been increased. We are comparing them. So, in this case, who is our nominator here? Is protozoa's count. The denominator is naive's count. So if it is negative, it means protozoa has less of that certain compared to naive. And if it's more, protozoa has intuitively more of that, okay? This is fold change. We log to it so that have a clear demonstration. And then here is the name. Right with me.
I have a question. If it's similar to the RNA sequencing data previously I was working, so protozoa here is like the housekeeping genes. Then we have something like the default, and we wanted to compare it to that default. We know the results for that species or
precisely, yes. So in the gene expression here, each of these are one of your genes, okay? In your expression data, and this is here. If it's about a sample, if it's about a segment or a cell, this is going to be this cell against this cell, okay? So you compare that, and remember that's why I said the order of this guy up there is important because you need to know who is going to be your numerator, who is going to be your denominator, okay? And then here you can also paint it with other, how do you call it, lower taxonomy level, okay? With me. You don't have to do that. And then you can also increase this tick number or decrease it, but just stick to the default. If it's too condensed, you can make it a little low or less. Anyway, it doesn't respond. Okay, yeah, cool thing is, whatever you see here is basically significant changes, adjusted for family-wise error, okay? FDR, by Benjamin Hochbier. So everything you see here is significant. And you can also see the data here that we are looking for adjusted p-value. Adjusted p-value is basically significant for this alpha level. And now we are going to association plots. Again, relative abundance, not absolute, we don't have it. Let's stick to genus. And then here it detects all the numerical variables in your data. Like, again, this is a dummy variable, I made it myself. It didn't exist in the data. We have short-chain fatty acid expression of some genes, body mass index, weight, blood pressure, and diabetes index, okay? And I looked into Google what is the range of them, so it's not very often. You select all or select only one of them, so all of them have been selected. Then you can choose Spearman or Pearson correlation. Then here you can see the number of most important taxa for the association plot. And then down here, color, whatever. Let's just, and then you can also bring some information because this is genus. You can also bring some information of family and phylum and kingdom into thing. Scratch this. This is completely redundant. Don't touch this. Nothing happens. And do you want to cluster the rows, which are your numerical variables? Yeah, I want to cluster. I want to see if, for example, body mass index clusters with blood pressure or not. And then give me the number of the cuts. This you can play later. Just let's go for 4. And then this is for taxa. Okay, and then I say, should I cluster rows? Yes. Should I cluster columns? Yes. And now, bam! You should have a beautiful heat map of your association, like that. And then I'll tell you how to read it. If you also have a chance to overflow it on the x-axis. You see here this cut, diabetes index, body mass index, and weight. Although it's not significant, otherwise you would have a star sign on it. They have been actually clustered together, and that's cool. You see, short-chain fatty acid doesn't have anything to do with this. And you can also see this clustering of this different taxa up there, belonging to phylum, kingdom, phylum, family. And these are your legends, like this. You can also define which color group do you like. It's actually quite cool. Let's go for red, and then recreate the plot. Like this.
Which underlying package are you using to make this one?
A few of them actually.
Okay.
Feed map is one of the packages for clustering. We are using HClust, thank you. The color, I wrote a function myself to create a discrete color, so on and so forth. But you don't need to be worried about that Magia. And then you can also download this table and do more thing about it. That's it. We go to phylogeny. If you have tree in your data and you want to actually also see how your variables look with the phylogeny. Let's say that I want to visualize the phylogenetic tree at genus level or species, whatever. Let's just do it. See what happens, okay? It should give you a basic tree. Needless to say, you have a lot of different ways of visualizing your tree, okay? But let's just continue with this circular. Maybe it's easier to follow, okay? You can align the branch or decide not to, right? You see the clades, how they are standing next to each other, how distant they are from each other. Who are these clades? We call this clade, okay? So this is the nucleotide. I mean, this has been some weights in it. It's not just nucleotide, but the nucleotide differences have been translated. I think it's based on average nucleotide identity. Have been translated to some weights, and then the longest they are from each other, they are more distant, and the closely related species or genera, they are on the same place. Okay, and now we want to bring a little bit more information into this. You can see that, okay, which phylum do they belong to? Say, or maybe you could also say, I want to highlight some of these clades that are important for me. I don't know, some random thing. And then you want to add some tip point that's just for visualization purposes. Here, taxa level for the color. Let's say phylum. This is beautiful. You can see that how the different phylum, they are basically like Bacterodota, for example, on the same clades with the same most common, recent common ancestor probably. You can also add that, I believe. Yeah, highlight the most common recent ancestor. That's it. So ugly. This line. Right? Better. Spend a little bit more time to read it, but better. You know what I mean? You cannot immediately see the distance, but when you look a little bit deeper, You see how they're distant from each other. And the clade is very intuitive, it tells you. What else you could do here? Branch length. This is a little bit graphically expensive because, sorry, computationally expensive because it's using a bootstrap to calculate this distance, okay? So, but voila, you have it here. Sadly, it's not responsive. I couldn't figure out how to do that. So oh yeah, here, this one. You can also bring into account the abundance information depending on your treatment, for example, in this case, or week, whatever, and see how the clades are related to, for example, this naive or protozoa in this case. But this one is a raw count. No, it's a relative abundance. You can also transfer it into log abundance or absolute abundance. You see things are getting a little more better resolutions. You can also increase this circle around the heat map around your pan tree, right? Just heat map distance. Yes, that's why I remember I had an issue when I was downloading it. That's why I added this function to it. Let's go a little bit higher. If you have long names, like for species, increase this as much as you can, okay? And then let's download it. There we go. Actually, it's readable, those numbers. But still increase it. I mean, the limit is the CPU, not the sky. But you get the point, right? Now you could also play with it. I'm done. Thank you for your attention. This was Microloop.
Nice.
Yes.